Hi, welcome back to My Smart Learning. This is a lesson for Year 12s and Year 13s for your A-Level Biology. It's chapter 12 from the Caboodle textbook. We're looking at respiration. Now, before we start looking at the first lesson, respiration on glycolysis, this is our learning objective. How do I release energy to survive? We need to do our quick recall quiz, our high five for our virtual high five. Number one, what are the products of the light-dependent reaction? Number two, what are the products of the light-independent reaction? Number three, what are the four main limiting factors for photosynthesis? Number four, what do the graphs look like for those limiting factors? And number five, what is the word and symbol equation for respiration? That's your sort of GCSE question as a link into our new topic. So, press pause, have a go at answering those questions, and once you're ready, if you unpause them, we'll go through the answers now. Right, so, what are the products of the light-dependent reaction? Well, if you remember, water goes in and light hits that water molecule and you get a process known as photolysis, which splits the water into an oxygen molecule, but you also get hydrogen and you get electrons. So you get protons, which is the hydrogen ions, and you get electrons. But that's not the end of the story. What are the actual products though? That hydrogen joins on to NADP, so it becomes NADPH, also known as reduced NADP. And with the electron, that goes into electron carriers, that comes down, and that energy is transferred to make ATP. So the two products then for question one are ATP and reduced NADP. Now, question two, what are the products of the light-independent reaction? Well, remember you got ribulose bisphosphate. So this is our Krebs cycle. You got ribulose bisphosphate, which is your 5-carbon molecule. Carbon dioxide joins onto that. You end up with 2 lots of glycerate 3-phosphate, GP. That then gets turned into your triose phosphate, your 2 lots of TP. That then will eventually go back into making ribulose bisphosphate, so your 5-carbon RuBP. Those triose phosphates then get converted into hexose sugars eventually. Now, what are the 4 main limiting factors? You've got light intensity, carbon dioxide, temperature, but you've also got the amount of chlorophyll or the amount of chloroplast that you've got there. What do the graphs look like? Well, if you've got the carbon dioxide or the light intensity graphs, they go up and then they plateau because you can't go any faster because you've got maximum amount of CO2 or light energy for the light intensity. Same thing for chlorophyll or chloroplast. They'll go up and they go flat, plateau, because you can't have any more chloroplast. On the other one though, the temperature one, it goes up, it hits the optimum, and then it goes straight back down because the enzymes denature and the plant will eventually die. And the last question, number 5, linking today's lesson, respiration. If you remember from your GCSEs, respiration is the release of energy from your food. It's not breathing. That's a common misconception people get with the word respiration. Respiration is about the release of energy from the food that you've eaten. But obviously, you need to breathe in to get the oxygen, to get the aerobic respiration to occur so that you can release the energy from that. So the word equation is glucose plus oxygen gives you carbon dioxide plus water. And if you want to balance that simple equation, I call it the devil's equation. 6 CO2s plus 6 H2Os gives you, oh sorry, my bad, that's photosynthesis. We're looking at glucose plus oxygen gives you carbon dioxide plus water, that's respiration. I was going on to photosynthesis there, and if you remember, they're back to front, they're flipped back to front. So here, glucose starts with C6H12O6 plus, this is the devil's bit, 6 oxygen, so 6 O2, then an arrow gives you 6 carbon dioxide, 6 CO2 plus 6 H2O. So if you remember the products, you breathe out onto a mirror, carbon dioxide comes out, but you get water vapor. That's your 6 carbon dioxides and your 6 H2Os. So what do I need to know from today? Respiration. We're looking at the first stage of aerobic respiration, and this is known as glycolysis. you need to know or outline what happens in glycolysis and how does it fit into the overall process of respiration. We need to describe the main stages of glycolysis and its products. But before I go on to that, let's go back to the GCSE content, the basics of it. Well, where do you get your energy from? Well, what you should know so far from GCSEs is that the energy gets released from food sources. So glucose is the most obvious one, because you do get other substrates as well. But the one that you get taught up to GCSE is glucose is the thing that releases energy and gives you energy to do stuff. So what is that energy needed for? Why do we need energy to survive? Have a think about that. So all of your life processes, every single one. So what are the 7 life processes for all living things to survive? Now you might have learnt it as Mrs. Gren, or when I was at school we learnt it as sort of Phlegma, or it depends on your textbook, you might have said Mrs. Nerg. But those first letters of Mrs. Gren, for instance, represents the first letter of that particular process. All living things must do all 7 of these life processes to be classified as a living thing. So M for Mrs., M for movement. So to move, you require energy. R for respiration. Now, ironically, respiration is the thing that releases energy, but you can't release energy unless you put a little bit of energy in the first place. So you actually need a little bit of energy to release the energy. So respiration. S stands for sensitivity. So all living things need to be sensitive to their surroundings so they can respond to it and adapt and move and go towards things that are favourable to them and away from danger. The G stands for growth. All living things need to go from baby size to adult size. The R for reproduction. So all living things need to reproduce to bring offspring, and then the offspring need to grow into adult size. E stands for excretion. All living things need to excrete and get rid of their waste products. And N stands for nutrition. All living things need to feed on a food source. Okay, so respiration is the energy that gets released from our food, but what's it used for? Well, it's used for every single life process. So it's not just for moving. It's not just for keeping warm, because we're warm-blooded organisms. You need it for every single life process. Even when you're asleep, respiration is required. So when does respiration happen? Happens all the time, 24/7, 7 days a week, every day of the year. Whereas plants photosynthesize only during daytime. So plants, when do they respire? All the time, 24/7. So plants are respiring the entire time of their life, because once you stop respiring, you're dead. Okay, so that's what respiration is in a nutshell. Now, the thing about respiration is it happens in small, manageable stages, because ultimately we end up with a compound called ATP. And I'm not talking about 20p short of a pound. No, you don't get that. ATP, no. ATP, adenosine triphosphate, is the chemical that has energy in there, and as it hydrolyzes and splits, it becomes ADP plus PI. Can you remember from chapter 2? The inorganic phosphate, there's energy in that bond. And you're transferring the energy from the respiring, the biochemistry you look at, from your glucose molecule. The energy in there is being transferred into those ATP molecules, and those ATP molecules are small manageable packets of energy that are soluble, and you can turn into ADP plus PI, but it's a reversible reaction. It can be like a jack in the box. You can press it back down, you flick the switch, it'll pop back up, you can press it back down, and ATP, ADP plus PI is that reversible reaction, and you store energy in that little bond. And that energy is what we need for all of those life processes, but it's small manageable amounts. If you think about glucose, we don't release the energy from glucose in one go. If you did, you'd burst into flames, okay? Well, not literally, you'd overheat, because there'd be too much energy released in too short a time, in a very small time span, and therefore you would overheat, which will denature your enzymes and you would eventually die. So energy needs to be released in small, manageable amounts. And this is why respiration happens in stages. And we're going to learn about these stages over the next 3 lessons. And we're looking at today's first stage, which is glycolysis. Now, what would it look like if you released the energy in one go? Well, you may have seen the Screaming Jelly Baby demonstration back at school. If you haven't seen that one before, please make sure when we're back at school that you ask me, and we'll get a lab sorted, and I'll show you the screaming jelly baby experiment, showing you the amount of energy in one jelly baby. If I heat it up with some oxidizing agent, you'll see the whole thing just burst into flames and you get this massive amount of smoke coming out. Showing if you release energy too quickly, you might get loads of it, but it'll happen too quick. You get an overheating effect. So with aerobic respiration, it happens in small steps. These steps generate small amounts of energy. So I use the analogy like this. I think I've already spoke to you guys about this when I taught you about ATP a long time ago. About if it was your birthday, for instance, and I gave you a £50 note, you'd be very happy. I'm assuming you'd be very happy. That'd be very generous, £50. Now, if you imagine that £50 note, if I said to you, go and spend it now, go into the shop, you know, you'd probably just blow it all in one go and go crazy. I don't know what you'd buy. I don't know what your kids buy these days. Maybe Chupa Chups, lollipops. I don't know what you kids eat these days, right? Collie cubes, whatever. So you go crazy. You buy all the sweets in the shop, all gone, and you've eaten it all and mess and sticky mess everywhere, right? Or you might want Krispy Kreme donuts or something. You just blown it 50, or you bought 10. Those are really expensive donuts. You probably buy 10 of them or something, and they all gone, right? You've had a bun fight, but that would release all of that money gone quickly in one go. Okay. Now, if you imagine, I say to you, I'm not going to give you £50 note, but I'm going to still give you £50. I'm going to give you in £1 coins, and every time I say, right, you can go and spend that money now. But I don't want you to spend it in one go with £150 to the checkout. I want you to go to the checkout 50 times, because you can only spend £1 at a time. So you have to keep going to the cashier every time. So you'll have to go to 50 visits and give them £1 to buy your donut. So that will take you a long time to keep going to the checkout and keep giving you £1 and getting your donut and eating it, right? So that's a bit like photo respiration. It's going in small manageable amounts. And you end up getting, instead of these £1, if I gave you that £50, you blow it one go, burnt it. Whereas if I give you those pounds, I can say, well, those pounds you can recycle. When you spend it, they'll give you back in 250p's. But then you can put the 250p's back together and you get £1. You can keep breaking them apart and putting them back together again and putting the energy back into there. And that's what's happening here. ATP is being generated. Now, the overview then. Today we're focusing on this bit of respiration. But if you notice, we've got two types of respiration. This bottom one here is aerobic respiration. So this is aerobic respiration, meaning aerobic and requiring oxygen. This is anaerobic respiration. Now they're both, anaerobic and aerobic respiration, have something in common, i.e. glycolysis. So glycolysis happens in the cytoplasm of the cells, because aerobic respiration happens inside the mitochondria. So this will happen in eukaryotic cells. These cells have to have mitochondria. Therefore, aerobic respiration can only happen in with cells with mitochondria in them, in eukaryotic cells. So prokaryotic cells can go through anaerobic respiration because there's no mitochondria. However, anaerobic respiration can also happen in eukaryotic cells when you can't get enough oxygen in. You end up going down that pathway. But that pathway is inefficient because it doesn't produce as much energy, as much ATP as the aerobic pathway does. The aerobic produces far more ATPs compared to the anaerobic respiration. So a quick overview then. The anaerobic respiration we're going to look at in the 4th lesson of the respiration sequence. So this week, in your 3 lessons, I'm going to do the aerobic respiration lessons. So today I'm looking at glycolysis. Our next lesson, I'm going to look at these 2 bits called the link reaction and the Krebs cycle. And in the last lesson of the aerobic respiration section, I'm going to look at oxidative phosphorylation, which is also known as the electron transport chain. So that's that bit there. That's the 3rd lesson. And in the 4th lesson, I'll look at fermentation. So glycolysis, a quick, very simplified overview of it. What do I get from glycolysis? My glucose molecule turns into this. It turns into pyruvate. I need pyruvate. Then what happens to that pyruvate? Either goes down the anaerobic route, which becomes, in animals, it'll become lactate, and then eventually lactic acid. But in plants and yeast, it'll become ethanol and carbon dioxide. However, when pyruvate enters the mitochondria, it will become In the link reaction, the main product is acetyl coenzyme A. That acetyl coenzyme A will enter the Krebs cycle, and then the products of the Krebs cycle, which are the hydrogens that we need and the electrons, they will enter this electron transport chain, and then this is the end of the respiration, because you've got oxygen there is the final electron acceptor. The oxygen is what turns into water there. So today then, stage of glycolysis and what's the yield? What am I getting out of this? So glycolysis, the first stage, this is happening in the cytoplasm of the cell. The first stage is this. You got your glucose molecule you got from your diet, something you've eaten. And the first bit, this is known as substrate level phosphorylation. Substrate level phosphorylation. So what does that mean? I'm making ATPs not from redox reactions, but from the substrate level, from the substrate itself. Now, the glucose molecule then becomes activated. So it says activation of glucose by phosphorylation. How? Well, ATP, I need to use 2 ATPs to join onto, or to release the inorganic phosphate, to join onto those 2 ends of the glucose molecule. So here's my glucose. What's happening is ATP, and ATP is going to hydrolyze. So energy is going to be transferred, and in that hydrolyzed reaction, I got inorganic phosphates because I got ADP plus PI. Those PIs will join to the end of the glucose molecule. Therefore, this glucose molecule now is called phosphorylated glucose because it's got phosphate at the end of it. Now, what will happen is, imagine there is like a biscuit, like a Kit Kat or something, where I'm going to hold either end of it on those inorganic phosphates, and it's been energized. I can split it. I'm going to twist it and snap it in half, and that's what happens here. I'm going to snap this phosphorylated glucose in half because I've got 2 little handles now to do it with. I'm going to split the phosphorylated glucose, and I end up with a compound that you've probably heard of from photosynthesis, my triose phosphate molecule. Remember my TP? There you go. I've got my 2 triose phosphates, and because one glucose molecule can make 2 triose phosphate molecules, therefore I've got 2 little flow charts there going on, little assembly lines. Each triose phosphate then goes through oxidation. Now, if you can remember from oxidation is the loss of electrons, reduction is the gain of electrons, but oxidation is also the adding of oxygen, and therefore reduction is the removal of oxygen. But there's one last one you need to know of. Oxidation is the removal of hydrogen, and reduction is the gaining of hydrogen. So if you look, notice here, this TP, the triose phosphate, it's being oxidized. It's being oxidized because it's losing hydrogen. So each of those TPs, the triose phosphate, loses 2 hydrogen atoms there. And what these hydrogens do is they join onto this coenzyme here, which I'm going to talk more about next lesson, this NAD, and it becomes reduced NAD. It becomes a reduced coenzyme. So NAD gains 2 hydrogen atoms there. Remember, reduction is the gain of hydrogen. So this NAD has gained hydrogen, so it's been reduced. So now I've got this reduced NAD. Now, what's the point of the reduced NAD? Well, reduced NAD is really important because it joins onto, really important for this bit here, the oxidative phosphorylation, because that's where it gets the hydrogens from. So these hydrogens here will end up in there. Well, that's where these guys come in. So this we'll come back to these guys next lesson and in the last lesson and explain what these guys do. The NAD then becomes reduced because it gains hydrogen from the triose phosphate. But what also happens? This process then converts 2 ADPs into 2 ATPs down one side. But how many ATPs did I get? How many triose phosphates did I get? I got 2 triose phosphates. So therefore, everything that's happening on this side also happens down this side. So, triose phosphate loses 2 hydrogen atoms, reduces NAD, and this changing also produces ATP. each triose phosphate will give me 2 ATPs, and once it's done that, you end up with a compound called pyruvate. And that's the really important thing I need at the end of this stage. So when we looked at this, glucose becomes pyruvate. It's not as simple as that. Glucose becomes pyruvate. In this lesson, you need to learn this flow diagram. You need to memorize the stages and the sequence of events that's happening here. So you need to draw this out, and by copying it first, by visually seeing it, then try to get it into your memory, and then try to draw it, maybe partially look at it and draw the first half, and then try to draw the second half with it, looking at it, and then try to draw the whole thing again without looking at it. Because these flow diagrams that you're going to see that come up in photosynthesis, that are coming in the biochemistry and respiration, you need to memorize those sequence of events. So, glucose becomes pyruvate via, first stage, the activation of the glucose by phosphorylation. That splits the phosphorylated glucose into 2 TPs. Those TPs then lose 2 hydrogen molecules to reduce NAD to become reduced NAD. You get a couple of ATPs made in that process, and you eventually end up with pyruvate. But what's the yield? What am I getting out of this? Well, remember what I said about you got to put money in to make money? I didn't use those phrases. I used you got to use energy to make energy. Like, as I said, what's the point of respiration? Like, you know, what's the energy used for from respiration? Well, you need it for respiration. Why? Because actually you need to use some energy first before you actually get some energy out. So here I've used 2 ATPs, but how many did I get at the end of it? I got 4. Got 1 there, 1 there, 1 there, 1 there. So the net, or the yield of ATP is 2. Because you got 4, take away 2 there, so you've got 2 overall that are extra. So the yield for ATP is 2. But you also get a reduced NAD and a reduced NAD. So you end up with 2 lots of reduced NAD, which is going to end up here. So you're going to end up with lots of ATPs from them too as well. So ultimately, you do get extra energy. But it's not very that much efficient, because you're putting 2 in, you're only getting 4, so you're only getting 2 on top. If this was anaerobic respiration, there would be no oxidative phosphorylation, there would be no electron transport chain, which means you'd only get 2. So it's very inefficient. You're only getting 2 for every glucose molecule. So not that efficient. Therefore, energy, that's why lactic acid builds up, and you eventually your legs cramp when you're going for your run, and you're going to eventually collapse. Okay, so anaerobic respiration ain't that great, but it's needed for survival if you haven't got enough oxygen and you've got an oxygen debt. So that's the end of glycolysis. There's some summary questions for you guys to have a go at. Look in your caboodle textbook, and the answers are there too. Hopefully you've learnt what glycolysis is in the first stage of respiration. If you got all 5 questions right, then you get a virtual high five from me, and I'll see you guys next time. A virtual high five from me.